Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about integument or skin. We want to gain a greater appreciation for the diversity of functions of skin and to recognize the cells and structures of skin that make possible this functional diversity. If this information is useful to you, please share it with your friends and colleagues. Thank you. Skin or integument. Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about integument. And here we see skin. This is a dermis and the epidermis, dermis. You can see the interactions between the two. You can see hair follicles here, fingerprints. All that relates to skin and their products. We want to gain a greater appreciation for the diversity of functions of skin and to recognize the different cell types and structures of skin that makes possible that functional diversity. Now, what are the functions of skin? What are the diverse functions of skin? Here we can see in body whorls, we can see where the skin has been removed from this individual, and you see that skin is fairly thick. It's the largest organ of the body. It protects against injury and desiccation. And here you can see the little boy has noted an injury. It also maintains water balance, excretes substances, and thermal regulation. And also it receives stimuli, temperature, pain, as the little boy noting, pressure. It also is a basis of recognition and yields clues about one's health. It doesn't take much effort to realize which one of these are the mother and which one is the daughter. So fat metabolism in a subcutaneous layer. So underneath there, so this is the epidermis, dermis. This is a subcutaneous layer which has the fat in there. And as I mentioned, there's various diverse functions, and there are different components of the skin that makes possible these functions, including we'll learn about how desiccation prevention is maintained in the skin. So here we see a chunk of skin. You can see the epidermis at the top, with the epidermis projecting down into the hair follicles. The dermis is a thick portion in through there, and then you have the hypodermis, which has the fat cells located in here and connective tissue. If you would take a rat and skin him and pull him back, you can see the innards inside there, but what's layer outside there is a skin. We're looking at the subcutaneous layer, which has fat in through there, and there would be the dermis and epidermis above that. Here we see with body whorls, you can see skin on the outside in through there, which is the epidermis and the dermis connective tissue that we see, epidermis and dermis. We can see it again. This is an individual that has a lot of fat in him. You can see the fat located in the hypodermis or just below the epidermis, the dermis. So obesity relates to too much fat in the subcutaneous layer. And we can see these two individuals. There's one individual, these are body whorls have these, 120 pounds. You can see there's not much fat there. However, there still is some fat here. Higher concentrations in a certain place, like in the body area. But this individual is over 300 pounds, and you can see the fat, the thick layer there, including humped up back fat on this individual, much thicker than even this individual here. So the skin has a binary origin. The epidermis comes from the ectoderm, but the dermis comes from the mesoderm. So the connective tissue below, the part that gives the thickness to your belt, is the dermis, and it is from the mesoderm. So there's different layers of the epidermis, the outer layer. You got the stratum basale. This is where the mitotic activity occurs to replace these other cells that differentiate as they move to the surface. You have the stratum spinosum, and their spines because they have desmosomal connections in through there. And when the tissues shrink, the desmosomes hold tight. It looks like they're spines, but really it's just a desmosomal connections of one cell attached to another cell. And these desmosomes continue all the way out to ultimately they're going to be sloughed off. Stratum granulosum is where the layer where you actually have granules in through there. There's keratin hyaline granules. And then the stratum corneum is the outer layer that we see in through there. So basically the cells divide by mitosis on the base, and then they differentiate as they go to the surface. The nucleus flattens, ultimately the nucleus is removed. Organelle content is digested with just the keratin hyaline granules making the matrix of the stratum corneum. So we can see the different layers again. Stratum dermovativum or basale in through there. That's a layer that touches the dermis. This is the epidermis, dermis. And then there's stratum spinosum, stratum granulosum. If you got real thick skin, you also have a stratum lucidum, a kind of clear area here that gives some cushion to it. And then a thick stratum corneum that we see there. So in the stratum corneum, you have the keratin hyaline granules, they're flattened, denucleated dead cells up through there, and they have differentiated from the surface. Stratum granulosum is where you have keratin hyaline granules that you can see. Stratum spinosum is where you have desmosomes and tonofilaments there, and the stratum basale is where you have the layer that's continually dividing. So you can see the stratum basale actually touches the dermis. Dermis, you can see here the collagen bundles, or longitudinal view and cross-section of them, and you have a basal lamina. The basal lamina is at the base of the epithelial cells that touch the dermis that we see. And so we can see the different layers, and then the stratum spinosum, you have the spine. These are the desmosomes and the tonofilaments that's associated with those, and that gives you kind of like a prickle appearance as the desmosomes are holding tight. As you move on up, you can still see those spines when the cytoplasm of the cells kind of pull apart, but the desmosomes still hold tight. And here we can see the different layers. You can see the spines right there very nicely of the stratum spinosum, stratum basale, and through there, this is the epidermis and keratin hyaline granules that we're seeing there. So as you move on from the stratum granulosum, where you have keratin hyaline granules, ultimately cells deteriorate further, organelles are in, and you just have the keratin hyaline granules. But note that even these cells are still attached by the desmosomal connections that we saw early on. So stratum corneum, denucleated cells, cell organelles have digested, and only the keratin hyaline granules remain. Here we can see the keratin hyaline that's inside there, and you can see desmosomal connections as the cells go all the way to the top to ultimately be sloughed off. But here we can see in thick skin the cells that are there: stratum corneum, stratum granulosum, stratum spinosum, and the stratum basale. There's three types of granules that are located in keratinocytes. Keratinocytes is the one that makes up the epidermis. There are some other cell types in there we'll talk about. And there's skin pigment. Melanin is the pigment. That's what gives it its color. So melanin granules are actually produced by the melanocytes, a special cell in the skin, and they're passed on to the keratinocytes by a cytokine secretion. So melanin granules is one of the granules that gives you color. There's also membrane-coating granules or lamellar granules, and those are waterproofing. Remember, one of the functions of skin is to prevent dehydration or desiccation. And so the membrane-coating granules discharge right in the stratum granulosum, and that cements the different cells, and that makes us waterproof. so to speak. So waterproofing function, and it's produced by the keratinocytes. So there's also keratinohyalin granules. Here we see the keratinohyalin granules that are concentrated, and they're mostly desmos, a ton of filaments associated with them, and then intermediate filaments, and then they become the matrix of the stratum corneum. So there's three different types of granules, and the granules do different things. One, making the skin resistant, or preventing desiccation, or giving color to skin. The different granules have different functions that we can see. Now the keratinohyalin granules that we see there, rich on histidine forms, matrix of cells stable due to a lot of disulfide bonds absent in hair and nails, oddly enough. But this is very stable. You can put your hand in gasoline and it won't dissolve, because these keratinohyalin granules are very insoluble, largely rendered by the disulfide bonds in here. So here we see the keratinocytes that are located in through there, the basal layer of stratum spinosum, stratum granulosum, right into the stratum corneum at the very top. And here we can see melanin capping. So melanin granules occurs right above the nuclei of these cells. In doing so, the granules stand between the cells that have the capability of dividing and the ultraviolet light produced by the sun. And so here we can see the melanocyte. Here's a melanocyte right in through there that's producing melanin granules, and that would melanin granules go from the melanocytes to the keratinocyte, and here you can see where the granules are capping. This is the same that we see there, so that you have the melanin granules that are above the nucleus to prevent DNA damage of the cells. Here we can see the capping very nicely here. The sun will be up through there, and it's only these cells in the base that they're concerned about. You don't really see melanin granules in these other cells. As they differentiate, they lose their content. And here we see the melanocytes is the one that produces the clear cell. It doesn't make desmosomal connections, so when the tissue is processed, the cytoplasm shrinks, and you can see it as a clear cell. So a clear cell in the stratum basale is likely to be a melanocyte that produced the melanin granules that then passed on to the keratinocytes. We actually see it on the keratinocyte. If you take the epidermis and pull it off and look at that, then what you will see here is the epidermis. So this is the bottom of the epidermis as seen by the dermis. And here we can see the melanocytes, and you see they're dendritic. They have branches that run out through there, and those branches help distribute the melanin granules to the keratinocytes. There is differences in thickness of skin. You could have it very thick, like a sole of a foot, 1.5 millimeters in thickness. You can have it very thin, like an eyelid. Also, it can be transparent, like the cornea of the eye. You can see we're looking through the cornea of eyes all the time. And skin can have appendages. One of those would be hair follicles, nails, glands, these different ones. And here we can see the skin on the ears. Sole of a foot is very thick there, dense, thick skin, thin skin. Trunk skin, and the tongue is another like skin. However, it doesn't have the stratum corneum on the top. So here we can see a thin skin is usually referred to the epidermis that is thin versus a thick epidermis that we see there. There are cells in the epidermis, different ones. There are keratinocytes is the main cell that we see. There's also melanocyte we talked about that produce pigmentation. That's from the neural crest. There's Langerhans cells that has the immunologic role, and there's Merker cells associated with nerves. And here we see a Merker cell associated with it. They still make desmosomal connections between adjacent cells, but you can see those in the basal. So the keratinocytes are the one that continue, renews it, and then these other cells are these same cell types differentiates. And this in addition to having the melanocytes that produce pigment. Langerhans is associated with limnologic barrier and the Merkel cells. So even the different cells that's in addition to the keratinocyte that's in the epidermis all have different functions. Here we see a melanocyte with its dendritic process, and you can see the cytokine secretion where the melanin granules are discharged into the keratinocytes. One cell produces it, but you normally see it in the other cell type. Here we see a little clinical correlation where you don't have pigmentation. If you don't have the tyrosine amino acid, then you are not going to be able to have pigmentation. This is the albino, basal and sarcoma cell carcinoma may develop. So a patient with albino would be more at risk to having a basal cell carcinoma and a squamous cell carcinoma because you don't have the pigment that prevent the UV light. Here we see a peacock. That's the albino. Freckles is another thing. Freckles is uneven distribution of melanin. It's in patches as opposed to an even distribution. In cattle, freeze branding of cattle, it kills the melanocytes, and so they do not pass the pigmentation on, and so it appears as white. Now remember the pituitary that we talked about before, and it's the melanocyte stimulating hormone that is produced in the pars intermedia, which is between the anterior and the posterior lobe. And here we can see the melanocytes at the bottom of the epidermis. Albinoism is failure to produce melanin, and that's what we see in albino rats. Malignant melanoma is a cancer of these cells. Addison disease, pigmentation of the skin due to adrenocortical insufficiency, and you have pigmentation positive in different groups, which is very similar to freckles. Langerhans cell that's located in stratum spinosum is responsive to gold chloride staining that you can see. Clear cells, no desmosomal connections, and it's a clear cell in the stratum spinosum. So a clear cell in a stratum basale would be a melanocyte. A clear cell in a stratum spinosum would be a Langerhans cell. And these have dendritic processes too, and they're associated with presenting antigens that might be coming through, pathogens that are coming through. Langerhans cell, they have a rod and racket. If you look at these, you look like the paddle of a tennis racket, and then the handles. This is characteristic of these cells, the shapes of the granules that they have. Contact allergic response, other cell-mediated responses, function immunologic role, antigen presenting is what it does. If we look at the epidermis-dermis interface, you can see it can be just slightly attached or very heavily attached. And this is what you have in the sole of your foot or the palm of the hand, where it's like this, where the epidermis could be pulled off of. And so you increase the surface area with the grooves and all, and that prevents the slipping of the two. If you would take the epidermis and peel it off the dermis, you would see little holes here which correspond to these projections. So these projections were in these holes there. And we can see how these projections of the epidermis was projecting down into the dermis. And here we can see even that the cells are attached at the base, are attached to the dermis. You can see the flaring out of the amplification of the cell membrane down here by the cytoplasm actually flowing out to increase the surface area for attraction of the basal to the dermis. Here again, we can see it again. This is the epidermis, stratum spinosum. We can see the spines. This is the stratum basale, which has these like flaring little fingers to allow the epidermis to be connected to the dermis. If you pull the epidermis off, you can see these projections here, these holes here is where the dermis would be attached. If you look at the finger pad, you can see there's a series of lines in through there that we will be going through there, and so this is the dermis looking up at the epidermis, and you can see how you have as the epidermis interacts with the dermal papillae, they will increase the surface area. So in the dermis, there's two layers. You have the papillary layer, that's the one that's scalloped, that interacts with the epidermis. You also have a reticular layer, that's the deep, dense, regular connective tissue that we see there. So these projections here is what we were seeing with the other ones. Here's holes here where sweat glands were coming through. We saw this before. There's some melanocytes located in there, and they're inside these projections. As we can see, this is a reedy pegs or dermal, and the dermal papillae would be what would be projecting up in there. So here we can see some skin. Epidermis is very thin, and it has smooth muscle. So smooth muscle can be seen in different areas. Aureoles, we can see it in the penis. We can see it in the peritoneal. We can see it in the scrotum, and this happened to be scrotum that we can see. The dermis can have skeletal muscle. As a rule, we don't have much skeletal muscle. Cow has a lot more. Horse has a lot more. They can move, control their skin shaking if a fly is on it. We don't have to do that, but we do have facial expression. And we can see a host of different facial expressions, and that due to your control of the skeletal muscle on your face. You can see a bunch of kids with their expressions, most of them happy, but some of them may not be so happy. So cutaneous appendages, you have the follicles, the pigmentation, and the graying is what we're going to talk about. These are the follicles here, epidermis, dermis, and the fat cells below. And we can see the graying that may occur. It will occur with age. And what happens is the melanocytes produce melanin, and it can be black or brown, kind of dark brown color. And also it can produce a reddish yellow color as well. When the keratin producing cells, the keratinocyte, die, they retain the coloring of it, so you see that. And so whenever you first go gray, the melanocytes are still present, but they become less active. So the melanocytes are there that produce the melanin, but they're less active. They don't produce as much. Less pigmentation is deposited in here, so it appears lighter, grayer. And that's the same thing as in the case of freeze branding of a cattle. You kill melanocytes, it doesn't put melanin granules in there, and it turns white. And here we can see the melanocytes are actually located in the bulb part of the hair, and they give the pigmentation that ultimately will be in the shaft of the hair. And here we can see that if you got straight hair, you have straight follicles, but if you got curly hair, you'll have curly follicles. And so you can either say straight hair or curly hair that you can see. And here we can see the cycle. You have hair grow, it's shed, and then another hair grows grows back routinely. Also, if you burn your skin down but you still have epithelium connected down below, then that epithelium will cover over the surface, as you see here and here, and you don't need a skin graft. But if you burn is below the epithelium, you'll need a skin graft to cover, because you don't have a source of epithelium that will populate it. In this case, this epithelium will repopulate the surface. And in fact, the depth has to do with the degree of burns, whether you burn below your epidermis or not. So here we can see a hair follicle. There are different parts of it. You got a sebaceous gland, which is what kind of oily secretion that makes our hair oily. We have an eccrine sweat gland, which discharges sweat on the surface of the dermis, of the epidermis. And then you have the apicrine sweat gland, and that discharges in the hair follicles itself. Usually, the apicrine sweat glands are associated with marking or something. So why not mark with a brush? And you can do that with the hair that we can see. Here we can see where the eccrine sweat gland has discharged sweat, and you can see it right on these ridges. In fact, these ridges are what gives you your fingerprints. So by having sweat right on the ridges, it leaves you're actually looking at the sweat in your fingerprints. So here we have scalp, which is very thin skin. You can see the epidermis there very thin, and then we can see the sebaceous glands that are located there, which is what we saw before with here, and we see the sweat glands, and these are the sweat glands and the sweat gland duct that ultimately will go to the surface. And so sebaceous gland is holocrine. The cell is a secretion. So when it's secreted, the whole cell is gone out. As opposed to eccrine sweat gland, that is like American secretion that occurs on. And here we see sweat glands have the myoepithelium, myo cells that surrounds each of these, makes a basket, and that squeezes out the liquid. Here we can see the pink, and here we can see individual cells making the kind of mesh, the myoepithelial cells or the eccrine sweat gland. So this is the secretory portion, this is the duct portion, and the duct portion is stratified cuboidal epithelium. I don't know anywhere else where you have that. Here we see the hair follicle and the sebaceous gland, and it's in the anal region that we actually see the apicrine sweat gland. They call it apicrine because they thought it was an apicrine secretion, like milk secretion. However, then they discovered it's American type secretion later on. And so we see the apicrine sweat gland. They have a bigger lumen in through there, and they're located at the anal region, so again for associating marking. There's also other types of glands. You got wax produced in the ear for waterproofing and to prevent insects from going in there. You have the tarsal glands that produces the tears, and the tears come in through there, and then ultimately they're discharged with lacrimal sac, lacrimal duct, and evaporated in your nasal breathing. And then you have, so here's the tarsal gland in through there, and this makes a kind of waxy substance on there, help hold the tears in. You know, if you take wax and put it around a glass, you can fill it up higher, and that's basically what it is. The secretions, waxy-like secretion, holds our tears in all the time. Mammary gland is another one of those glands that are produced by the epithelium on the surface from the epidermis, and here you can see where a milk secretion occurs by apicrine, and there's also American secretion for the casing. And it's the mammary gland for which our class is named Mammalia. It gives our name because of the mammary gland. Three things makes a mammal. One is milk, One is special ear bones and one is hair. We don't have feathers and we don't have scales. And see, as variation of microvasculature, you got the typical one is the arterial capillary venule. That's your typical way. But you may have a shunt that goes through there that bypasses the capillary bed. But in the case of skin, we have an AV shunt, a big shunt that opens up between the arterial and venous side, and that causes a plexus of veins to be amplified. And here you can see those cells there. Now, these are blood vessels that provide nutrients, but then here's a big plexus that when the AV shunt opens, the arterial blood runs into there. And as a consequence, that causes blushing. And that's what happens is you have a large flow of blood that's in the plexus below the skin, and you were able to see the blushing to occur. So as I mentioned, you have this shunt that causes blushing, and also it's associated with cooling of the body by letting warm blood up next to the skin. And so here we can see the projections in the dermal papillae. Remember, the epidermis doesn't have any blood vessels. This all comes from the dermis. So if we look at the blood volume, here we see a pie chart. Most of the blood is actually in veins, only 5% in the capillaries. And so the skin could have as much blood as your whole capillaries all over the body would have. And so the skin, normally a little flow occurs. However, on hot conditions, it may be a very rapid flow of blood coming through the AV shunt into these plexuses and then moving out. So blood vessels are in different layers to accommodate variations in rate of flow. So if we look at this is the epidermis here, the dermis. This is the reticular layer and the papillary layer in through here. Hypodermis, dermis, epidermis, hypodermis, where you have the fat cells. Also located here are the presenting corpuscles, and you can see them look a little bit like onion, and we see the eccrine sweat glands as well. Now there's epidermal, dermal nerve interfaces. You have free nerve endings, and there's different types of nerve endings. This is the bulb type nerve ending. And then you have the Merkur cell that we talked about before, associated with a nerve ending, one of the different cells of the epidermis. And they have sympathetic contacts with afferents that are coming out here. Light touch is what you see, and specialized coating afferent nerves resolve fine spatial details that occur. That's what it does, and that's in contrast to the Meissner's plexus, which is a nerve that actually runs up in the dermal papillae underneath the epidermis, and that's sensitive to light touch, and you have rapid adaptive physical deformation causes changes in capsule, causes you to be able to feel something, makes an action potential that stimulates a nerve. However, you quickly lose the sense to it, like you don't feel your clothes after a time. So it has adaptive capabilities, so you don't feel it the whole time. The Meissner corpuscles, the number of Meissner corpuscles per square meter in human and fingertips drops fourfold between the age of 12 and 50. So the number you have become less sensitive with age. And here we see the presenting corpuscle, which is a deep pressure receptor, and that transmits information about the pressure. And here we can see the different ones, free nerve endings, the Meissners, proscenium, and the different ones, and they respond to heat, cold, pain, touch, pressure. And that brings us to the mucocutaneous junctions. Mucocutaneous junctions. And there are different ones. One's your lips, your nose, eyelids, vulva, prepubes, anal canal. And since this is a large intestine, then this is the anal canal. It shows you these are simple columnar epithelium, all of a sudden becomes stratified squamous on the outside of keratin skin. So next time you see a big bunch of skin, I want you to think about the functional diversity. It protects you against injury and desiccation. We're able to walk on land because we have this capability. It maintains the water balance. It creates things, basis of recognition, and fat metabolism. We want to thank various books for some of the illustrations and some of the figures that we have here. They are the original sources of those and not me. There we see in Washington, D.C., they have a Holocaust museum, which I was able to go and see. All right, if this was useful to you, please share this information with your colleagues and your friends, and we'll talk next time. Thank you.